Because my grade is right like right on the cusp.
Yep, actually, I think I'm going to have everyone bring that back to my office.
Okay, guys. I know we're all so excited. It's the last day of class. I mean, so sad. If you guys want, you can come over winter break, sit here, and imagine me standing here. Listen, you can listen to my wife forever. That is on the internet.
I don't think anyone got hired.
Okay, did everyone just have an exam?
Yes.
So that's why there's that nervous buzz, huh? Advanced contract. I'm sorry, that's a hard class. So it's over. Very soon. Not over. It's not over. It's almost over. Okay, guys, let's try this. Okay. Come on, guys. Come on, keep it together. I know we're all losing our minds. It's okay. It's okay. We're going to be okay. Okay. This is the last lecture from me, and you don't have to worry about a test until Wednesday of next week. So you should all feel relieved there. Final exam is the 17th at 10:15 here. In this room. I've gotten so many questions. Is it in the computer lab? No, that's impossible. Too many of you. It's here. It's paper. It's going to be a ball. I'll try to bring donuts or something. You know, I'll pop up on sugar. Oh, for those of you who aren't taking the final, you can come take a donut.
Oh my God, yes. I'll leave them right here, and you can just come through the side entrance and get a donut. After class, I will check people's notebooks. My thinking is that I won't get done quickly today, so please just come to my office and show me your notebooks right after you have time, some point today. I don't want to keep them. I want you to be able to study with them. Yes?
So we can't come today or today?
Well, I'd like to get everyone's today. I don't want to keep them. I just want to look at them, acknowledge the fact that you participated, and give you your 5 points. And the reason I want them all now is so you can look at your grade book and know that you have your 5 points and what it does to your grade. Yes? Okay. There's going to be I just had a question on the final. Is there an essay on the final? No, it's going to be all multiple choice except for a bonus, which will require writing and thinking. Yeah.
Do you have an idea of how many questions?
100 million questions. There'll be like 20, 25 questions. I know you guys don't like that, but sorry, I'm not trying to make you sit here for 2 hours. I just want you to do boom, 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 done. Move on with your life. I know, but it's hard for me to come up with a million questions about such a small amount of content. It's not. Yes. I'll be around all day. Yeah, contrary to popular belief, it's like when you're in kindergarten and you think your teacher lives at your school, and I think when you're in college you think your teacher does nothing but stand in front of you and teach. I will be here all day, every day. All day, every day. I'll be here till 5. So there's a meeting at 3:30 that I have to be at, so ideal time to till 3:30. Okay, another question.
Yeah, what's the deal with the recitations quizzes since we didn't have?
We're dropping 1 recitation quiz.
There's no makeup as far as the points.
Huh?
There's no makeup as far as the points.
No. So your total points will be lower.
Okay.
It's still the same in the sense that your grade is dependent upon the percentage of possible points available. There are less points available because there are a few recitation quizzes that are. Yeah. Yes.
So were there only 6 or were there 7 recitations?
So there's one from last week that I still need to put in. I believe it's 6.
Okay.
I believe it's 6. Yeah, I always miscalculate the recitations. Okay, so exciting. Wow, we have so much energy this morning. So we're going to do our last writing thing. Okay, pull it together. We're all in it together. Okay, discuss the role of the ubiquitin proteasome system in cell cycle regulation. That's like a huge, you could probably write for pages. But the way to approach it is first think about the ubiquitin ligases and then what they target, right? There. Okay, guys, I'm feeling like people aren't into it. And so I'm not going to push it. I just have to go. Were you going to tell me people aren't into it? Okay, do you want me to come visit you? Okay, so finish writing what you can, and then when I come back down here, we'll go over it. Okay, guys, let's do this. So, I'm going to just let people raise their hands. So tell me, how did you approach this? Someone who feels good about life. No one's feeling good about life. Here, what is the ubiquitin proteasome system? I just said ABC. Okay, so that is one ubiquitin ligase we have talked about. Ubiquitin proteasome system means all things involved with adding ubiquitin to proteins that will be degraded ubiquitin. So APC, what else? SCF. Okay, so that's where your brain needed to go right away. APC, SCF. If you got there, then you've got like half the battle, right? And so then you needed to think, well, what do they degrade? Who can tell me something APC degrades? Yep. Ubiquitin, no. That's not weird. Geminin. Securin. Geminin, securin. Okay, so then you would make a list of things. So you've got geminin, securin, APC. What else? What else? That's because ATG degrades M-cyclin, which, yeah, M-cyclin, and then S-cyclin. Okay. And so what is SCF degraded that you guys learned about in class? Huh? SCF? What is SCF degrade? Anyone? Yes. Yeah, CKI. Yes.
Wait, so APC degrades
Cyclin, S-cyclin, Geminin.
and I thought that it's activated by
I know, isn't that weird?
Okay, so
That's that whole feedback loop that she was mentioning. So I just heard, wait, APC degrades M-cyclin, I thought it's activated by M-cyclin. Yes, it's a feedback loop, but then the way APC is kept on later is through CDH1. Okay, so then what you would do is you would go through each of those substrates and discuss what they do. Right, what does Geminin do? CDC1. Inhibits what? It's okay, so a Geminin basically, very broadly, is an inhibitor of ORC complex, origin recognition complex setup at the origins prior to S phase, right? So if you degrade the inhibitor, that allows to kind of set up and get ready for S phase. So that's one way the ubiquitin proteasome system regulates that by controlling presence of the inhibitor. I believe it's CBT1. So it's Geminin securin. What does securin do? It secures something. Thank you. It does. What does it secure? Separase. What is separase involved in? Yes. Uh-oh. Does APC degrade securin? Yes. APC degrades securin. Securin is an inhibitor of separase. Separase is involved in what? Sister chromatin separation. What is it separates? What is it cleave? Cohesins, right. So now you've got a way to think about it in terms of S phase. That's one role of ubiquitin proteasome system in S phase. One role of ubiquitin proteasome system in mitosis, sister chromosome segregation. And then what else we said? And then S cyclins, that's obvious to turn off MCDK. And MCDK, which is going to either allow exit from mitosis or prevent entry into S phase and prevent entry into mitosis until it's time. Okay. So that's how you would have approached that. Oh yeah, and then FCF, you said CKI degradation. What do CKIs do? Inhibit cyclin kinase. Yep. So exactly. That was this thing that bound to both cyclin and the CDK, prevented it from being active. And so then it has to be degraded in order for cyclin CDK to be active. I went to AFCB this weekend. I don't expect for you to know what that is. I know Michelle was there, kind of. She could find me. That's a American Society for Cell Biology conference. It was in Philadelphia. So it was an easy trip up. The woman who had a poster next to me was working on, this was so cool. I was like, you should come talk to my class. She was working on an inhibitor of an FCF complex. Inhibitor of an FCF complex that prevents degradation of a CKI to cause cell cycle arrest in cancer cells. She had gobs and gobs of data. It was beautiful. It was very cool. That whole conference, it's like the hugest conference ever, but it was like, there was like a huge cytoskeleton section that was like everything we've learned about, but like 100 times more detail. And then there was a huge cell cycle section, and I was like, I should have just told all of you guys to go to the conference instead of taking my class. There's a lot of good stuff for undergrads. So there's a lot of good mentoring stuff, CV review. It's really, you guys should look into, if you're interested in cell biology at all, I encourage you guys to look at becoming a member of that society, because stuff for undergrads is super cheap. Everything's really cheap. You can go to the conference for like $50. I mean, if you can get yourself there. Philly, that would be easy, right? Anywho. So far, this is what we've been doing. This is everything that we've done up to today. This is what you should be studying for the final, what we did on Thursday. We talked about regulation of M phase. Specifically, we talked about the mechanisms behind mitotic spindle assembly. Basically, what is MTDK phosphorylating and how does it affect M phase, right? Mechanisms behind mitotic spindle assembly, spindle checkpoint, that metaphase, anaphase transition, chromosome segregation, cytokinesis, the role of the cytoskeleton in all of this, and exit from M phase. Okay, so when we're talking about cell cycle control, we're talking about two sides of things, right? One is cell division, so stimulating entry to S phase, which then kind of starts the whole cycle, and that's stimulated by mitogens. An example of a mitogen is like a growth factor that might be secreted from another cell that's going to bind to a receptor and induce a signaling pathway that will result in transcription of S-cyclin, for example. So that's kind of what's happening right here. Cell cycle arrest is kind of the other side of things. So when things are going poorly in the cell, then you get cell cycle arrest. So if there's DNA damage or uncontrolled cell growth, something like that, then you're going to get activation of P53, which means it's going to be phosphorylated. It's not going to interact with MGM2, it's ubiquitin ligase, and it's going to activate transcription of proteins involved in cell cycle arrest and apoptosis. So think of it as kind of like two sides of cell cycle regulation, turning it on and turning it off. And that's it. No, just Okay, so mitogens, right? So these are these growth factors. We're going back to signaling here for a minute. So what they do is they stimulate cell division by stimulating G1-CDK and G1-F-CDK activity. So here's your mitogen, your signaling molecule binding to some kind of receptor, activating a signaling pathway. Here's a RAS MAP kinase pathway. This is why I say it's all kind of cumulative, isn't it? Because if you forgot what this stuff was, this isn't going to be as simple as it would be if you remember what it is. Because then you can go, oh, that makes sense. That's cool. So you get some kind of gene regulatory protein that's going to activate another gene regulatory protein, which will then activate G1-CDK. And then you get to the meat of it, which is you've got G1-CDK kinase, right? That's going to be active, so it can phosphorylate something. And it phosphorylates RB. And RB is an inhibitor of another transcription factor called E2F. And E2F will then activate transcription of S-phase genes, such as G1F and S-cyclins. When you buy your S-cyclins, you go into S-phase. When you go into S-phase, that's DNA replication, then you're going to go into M-phase, and that's what cell division is. That's what cell division is. So when you're taking another class and they talk about cell proliferation, they're pretty much talking about this kind of thing. So just to look a little bit more closely, your mitogen binds to a receptor, activates a signaling pathway. Here, RAS MAP kinase activates the gene regulatory protein, some kind of transcription factor, which will activate gene expression. So genes are being expressed. One of those genes will be MYC, which is another transcription factor. Anytime you have a transcription factor, it's going to bind to the DNA and activate transcription of something. And that's going to activate transcription of G1 cyclin to activate G1 CDK. So now you've got this active kinase that can phosphorylate RB. RB is an inhibitor of E2F. It keeps E2F off as long as it's bound to it. But when G1 CDK phosphorylates it, it turns into a mustache. Every single time I look at this, I see a mustache. Do you guys feel like hipsters are really into mustaches right now? Actually,
Mustaches are just coming back in.
they're coming back in and are there any mustaches here? Yeah. Yeah. You guys aren't sporting the hipster mustache. Sorry, I'm going to digress. I went to, because of Arby. I went to 930 Club the other week to see a band play, and my husband and I were remarking that in Baltimore, there's a different kind of it's kind of like a edgy mustache, where in DC, it's more of like a creepy, I have a van with tinted windows mustache. It was all over the place at the 930 Club. Okay, so, and I was thinking about RP and teaching you guys about RP while I was at the 930 Club. So you get phosphorylates, so G1-CDK is now active, it phosphorylates RB. So this liberates E2F. So now you've got this transcription factor that's active. And if you have an active transcription factor, what's it going to do? And it's going to induce some kind of gene expression. And it induces expression of S-phase genes, such as G1 S-cyclin and S-cyclin, right? And if you have S-cyclin, you can have what? S-phase, right, DNA replication. Right, so here's DNA synthesis. And that is how mitogens activate cell growth, cell division, okay, by stimulating S-phase. Okay, so when you're studying this and you're like, I don't even know how to think about this, you can look at these questions, right? How do mitogens promote cell cycle entry? Binding a receptor, activating a signaling pathway. Activating a signaling pathway that is involved with activation of transcription of S-phase genes. What role does RB play in cell cycle control, other than being your mustache? Yep. It inhibits E2F. It's an inhibitor, right? Okay, so then what is E2F? It's a promoter of cell cycle entry, but how? What does it do? It's a transcription factor involved in G1S or S-cyclin. Right. So you're studying a new protein, which you believe is involved in S-phase entry. You mutate it randomly and analyze the effect on cell cycle entry. You find that in cells containing your wild-type protein, so your non-mutated protein, 50% of the cells are BRD positive compared to cells containing the mutant protein, where with the mutant protein, 100% of cells are BRD positive. So what is a likely function of the protein? So I want you guys to take a minute to think about this as best as you can. And think to yourself, okay, so something that's normally inhibited is not going to be inhibited anymore, because suddenly you've got 100% of cells that are BRD positive. But you also have to ask yourself, what's BRDU? What does it do? What does it mean? Okay? And then I'll circulate and offer guidance if necessary.
No, there's like, no, that allows. Yeah.
Okay, guys. How's it going? Okay. So, who wants to take a stab at what the heck's going on here, or at least talk about how they approached it? Yes.
So we said that the new 13 would make SCP more active, therefore activating SCP more, so that's why you see more cells in SCP. So the wild type would inhibit SCP, and that's why you see 50% in SCP.
Okay, so that's one good observation. The mutant is making SCDK more active somehow, possibly through having more S-cyclin, which is why you have 100% of cells in S phase, because you are able to associate BRG positive with bleeding just on through S phase, and so that the wild type must be some sort of inhibitor. Okay, who went deeper than that? You said it had something to do with RB? Well, that's because you know my patterns. Yeah.
I said that the effect of the mutation having is that it's blocking RB, so when H is really active, so that's why you're 100%.
The effect of mutation is somehow blocking the function of RB, so E2F is staying on, so you have 100% BRG positive. So these are all the right ways to be thinking about things, right? So I'm saying, what's the likely function approach, and you got that. What effect does mutation have, and you got that. And really, this question was designed to make you think about RB. Now, the outcome of RB not working is going to be expression of S-cyclin and activation of SCDK, which is kind of where you guys in the back went directly. So that all of you guys together have the perfect answer. Any questions about this? Okay. Okay, so DNA damage is going to prevent entry into S phase. Does that make sense? Is it something you should memorize, or something that's just obvious? It's just obvious. Don't spend your time memorizing this sentence. Just don't do it. Don't write it on a flashcard. Right? Because if your DNA is damaged, you really should not be replicating it because chances are you won't be able to because maybe your double strand of DNA will be stuck together, won't be able to come apart, you'll have stalled replication forks, it will be a big mess. You don't want to do that. And so there are ways to prevent this from happening. So here, for example, showing you that when you have DNA damage, and here the source is X-rays, and it's going to activate a whole DNA damage detection system, which we are not talking about in this class. I teach it in 409. But basically, you've got these kinases that will be activated as in response to detection of DNA damage, and those kinases are going to phosphorylate P53. And when you phosphorylate P53, it prevents interaction with MDM2. That's its ubiquitin ligase that's constantly degrading it. And then active P53 is going to bind to DNA in the regulatory region, activate transcription of different genes, one of which is P21. And if you guys remember, P21 is a CKI. What's the effect of increased expression of P21? It's going to bind to and inhibit cyclin CDKs. Does that promote cell cycle or inhibit? Inhibit, right? Because you're increasing the presence of your inhibitor protein. Right, so you're promoting cell cycle arrest. Is that what you want to do if you have DNA damage? Yeah, if you promote cell cycle arrest, this gives time for repair. This prevents moving into S phase with damaged DNA and potentially creating either more damage or passing on damage to the daughter cells. So this is just close-up of this. So P53 levels are normally kept low by the MDM2 ubiquitin ligase, right? Now you should be able to relate this back to HPV a little bit. So this is the normal function. What did we do in recitation? HPV was actively degrading P53. So this kind of all makes sense here, right? So normally MDM2 keeps P53 low and keeps this whole response off. But with DNA damage, or other things, not just DNA damage, but other things that need to arrest cell cycle, you'll get phosphorylation of P53, that's the green one. And now MDM2 can't bind, it destroys the binding site. And it activates P53 so it could go into the nucleus and act as a transcription factor. And transcription factors activate transcription of genes. In this case, it's activating transcription of P21. In the case of HPV, infection itself was activating P53 and increasing levels of it. And that's not good for the virus. So that's why the virus had evolved a mechanism to degrade P53 on its own. So then you've got expression of P21, and now this CKI can inhibit the cyclin CDK complex and arrest cell cycle. Questions about that?
I just wanna, so if there's damage, then the P53 is gonna be phosphorylated. If there's not, then the MDM2 is gonna bind to it.
Right. If there's damage, then P53 will be phosphorylated. If there's no damage and everything is fine, MDM2 is always binding to it, ubiquitinating it, and setting it for degradation.
So the phosphorylation makes it the MDM2 go up.
Phosphorylation does, well, P53 is phosphorylated in a million different places in a million different ways. The most simplistic way to think about it is inhibition of, because one phosphorylation site inhibits interaction with MDM2.
Okay.
Yeah. Okay, any other questions? Okay, so you're studying P53 mutations. You find that a mutant form of P53 has a slightly lower or smaller molecular weight than wild-type P53 after exposure to X-rays. So cells are exposed with X-rays to activate DNA damage response. You should normally get phosphorylation of P53, but you're finding that P53 in this one mutant form is actually smaller after exposure to X-rays. So you do some experiments and you find that cells containing the mutant p53 have twice as much PI staining as wild-type cells after exposure to X-rays. What is a likely explanation for this? So you need to think about a few different things. One, how is p53 associated with any of this, right? What should be the effect of DNA damage or X-rays on p53? And then you need to think about how the protein's molecular weight might be affected. And you need to think about PI staining. What does that mean? Twice as much PI staining, what does that mean? So does anyone want to tell me just some thoughts that came to their mind as we were thinking about this? I see your hand go up back there. Who knows how the mutant became a mutant? But there's a mutant. Yeah. But the real question is what's going on here? Why is it smaller? What's happening? How's this affecting its function? How's this affecting the PI staining results? Yeah.
Is it that it's inherently, well, the mutant is not working properly, so it cannot
The mutant is not working properly and cannot arrest cell cycle. So that would explain twice as much PI staining because the cells are moving forward. What about the molecular weight piece? What does that mean to you? Yeah? Are you waiting? There's fewer p53, but p53 is 53 kilodalton. That's its molecular weight. But when you treat your cells with X-rays with wild type p53, it's a little bit bigger, whereas it's a little bit lower when it's mutated. Yep.
Is it getting phosphorylated when it's exposed to the X-rays, so that makes it like heavier?
Is it getting phosphorylated? Well, should it get phosphorylated when it's exposed to X-rays?
I would think so because it's damaged technically, and you're gonna want it when there's damage.
Right. Right. So wild type p53 is being phosphorylated after DNA damage because that's the whole way to activate it. But mutant p53, yeah.
So maybe
It's losing its phosphorylation site. Maybe the mutation is in its phosphorylation site. It can no longer be phosphorylated. It's going to be smaller because the phosphate group does make a protein heavier, right? Does that make sense? I didn't think about it that way before, but it is a covalent modification that will increase the weight of the protein. Can't be phosphorylated, and so now it's going to be degraded by MGM2 normally, and so then not activate transcription of a CKI or not induce cell cycle arrest. That's the way to think about this question. Any questions about that, the thinking process? Okay. Okay, so when DNA damage is too severe to be fixed, you're going to have cell cycle arrest or apoptosis. And so this is the PI staining that we've seen before, except for there's an extra peak you haven't seen before, right? So G0, G1, that's your one copy of DNA. G2, GM, that's your G2M, that's your two copies of DNA. Then there's this peak here that has way less than one copy of DNA. And those are your apoptotic cells, because part of apoptosis is that degradation of DNA, nucleic acid, right? So if you have excessive cell proliferation, so too much cell cycle going on, probably tumor cells in the neuron, then this will signal, then you'll have excessive MYC production, so a lot of MYC is being made. And so then what you'll get is ARF will be expressed. So MYC is going to induce expression of ARF, which is going to bind to and inhibit MDM2. So this is another mechanism of stabilizing P53. One mechanism is DNA damage, followed by phosphorylation of P53, which inhibits binding by MDM2. In this case, you're getting expression of a protein that is going to bind to MDM2 and release it from P53. And so now you've got a stable active P53 because it's no longer interacting with the ubiquitin ligase, and that results in cell cycle arrest apoptosis. So I have up here what's the normal role of MDM2, just to kind of remind you to think about that this is not independent from this last one we spoke about, right? It's not completely independent. It's the same system, just a different way it's being regulated, right? With MDM2 ubiquitin ligase. Anytime MDM2 loses interaction with P53, P53 is going to be stable. Questions about this? Okay, so I wanted to touch on apoptosis because we can't have a cell biology class without talking about apoptosis. It's always left till the end. I'm sorry about that. So I'm going to really test my new and improved memory by a movie of apoptosis. This movie was put out by a drug company, but it's cool. Ooh, nice. So we're going to watch it this way and see if I pass out. Are we having fun yet? Okay. Why does it do that, huh? That's what I want to know. Okay. Well, we're going to start this again.
Cell death may be triggered through DNA damage or severe cell stress.
Okay, so this is a really... Yeah, come back here.
Cells to avoid apoptosis.
Okay, guys. I know, this is so much fun. So this is talking about the two different pathways of apoptosis, the extrinsic and the intrinsic pathways, and I want you guys to understand that for the final. If you can activate apoptotic pathways, then you are good. So that's why it's a drug company that put this together. So this is talking about the intrinsic pathway, meaning something inside the cell is activating apoptosis. Here they're showing DNA damage is activating apoptosis. They're showing P53, and then we can talk about how P53 is associated with apoptosis. And basically what ends up happening is you get release of cytochrome C from the mitochondria, which activates the whole pathway system, and then you get apoptosis, all the membrane blooding and the degradation of every single kind of cell.
The extrinsic pathway is
Then there's also the
extrinsic
pathway of apoptosis.
This pathway
means
has the
stimulus is
potential
coming
to
from
induce different
outside
tumor
the cell.
cells independently.
And so here you've got fast and fast ligands
Development
are
of
a
novel
very common
molecules that promote apoptosis
example
by
for
targeting both pathways of
activation of the
apoptosis,
extrinsic
intrinsic
pathway,
and
and there's
extrinsic.
just another way of
Advances the
activating
understanding of the mechanisms behind tumor cell proliferation.
Well, I take that back. That was a horrible thing to say. But it's
Genentech
activation of pathways
Biooncology is
that can also
committed to
activate the
developing
intrinsic
novel
pathway.
molecules that promote apoptosis
And then this is then
by targeting the
advertising cell.
intrinsic and extrinsic
I had a friend
pathways.
who did some work with them. You know, there's a place to go get a job at. So when you're studying, feel free to watch that because I kind of like how it shows both different pathways nicely. I'm a big fan of movies. I know a lot of people are. In our last bit of fun, go through some. Oh, come on back. It'll come back. Look at that, guys. My computer did not crash. It's very exciting. Made it through. It's not getting hung up as much. Come on. So then I think this is an issue with this system more than my computer. My computer's just waiting. Okay. Okay, who loves apoptosis? There we go. Okay. Okay, so apoptosis and castrations would be kind of synonymous in your mind. There are two kinds of categories of castrations. Don't worry about all the numbers, really. Well, think about them. But you've got your initiator castrations, and those initiate the apoptotic event. And then executioner castrations that are going to directly cleave things, such as proteins, cytosolic proteins, nuclear lamina, all sorts of the things that need to be cleaved in order for apoptosis to happen. Right, so your executioner caspases, they target cytosolic endonuclease inhibitors. So an endonuclease degrades DNA normally. And so if you cleave an endonuclease inhibitor, then suddenly your endonucleases will become active. And so all your DNA is going to become fragmented. So that's one reason people look at DNA fragmentation as a readout for whether apoptosis has happened. Nuclear lamina, so that's your nuclear structure. So start to destroy your nuclear structure. Cytoskeletal proteins get targeted and cleaved, so this causes the cells to round up and start blebbing. Adhesion molecules, so these are those molecules like the ICAMs and that are involved in cells being attached to neighboring cells, so then they're going to detach from neighboring cells. So all of those things are cleaved, and that's what causes what you see with apoptosis. So here's your extrinsic pathway. So this is showing fast and fast ligand. And here we've got two different cells. Here you've got a killer T cell, and here you've got some kind of, let's say, infected target cell. And so FAS is the ligand, and then FAS is also a receptor. So FAS ligand is going to bind to the FAS death receptor. And then you're going to get these different adapter proteins that are going to bind and form this ZIS complex, which will then activate some caspases, some of these initiator caspases, which will then activate executioner caspases. The whole point is turning on activation of your initiator. So cells and viruses also include decoys that inhibit apoptosis, that are going to compete with these caspases. Okay, so then there's your intrinsic pathway of apoptosis. And that's going to trigger release of cytochrome C from your mitochondria. And this is usually activated by something inside the cell, such as DNA damage. That's a big one. So there's some sort of apoptotic stimulus. And then you're going to get release of cytochrome C from the intermembrane space of the mitochondria. And then it's going to bind to all these different proteins. That's going to form an apoptosome. Apoptosome, we like to own everything. Proteome, transcriptome, apoptosome. There's more, I'm sure. Who has more owned? Lysosome, nucleosome, everything. It's all owned. We're not very. Anyways, so you form this apoptosome, and this activates procaspase 9, which is one of these initiator caspases, and then that's going to cleave and activate the executioner caspases, and then you get that cascade. I think you can go back to that initial video I showed you. I think it does have some apoptosis in it from like day 1 of the class. So this is triggered from inside the cell, DNA damage, hypoxia, cell stress, and also a lack of extracellular survival signals. So there are signaling molecules. I don't know if I'll get to this, so I'll tell you about it. There are signaling molecules that will be secreted by other cells that bind to receptors on some cells, and if they receive these signaling molecules called survival factors, the cell survive. But if they don't get these signaling molecules, then they'll die. This happens with neurons sometimes. Okay. So, yeah, sure.
Are the cascades within the intrinsic pathway?
There's a cascade. That when I say cascade, what I'm talking about is initiator, activating, executioner, and that happens in all cases. It's just how that initiator gets activated. That's different. Right.
Okay.
Right. So let's go to this question, this practice question. The extrinsic pathway of apoptosis differs from the intrinsic pathway in that A, the extrinsic pathway is activated by cell surface receptors. The extrinsic pathway is acted by release of cytochrome C in the mitochondria. The extrinsic pathway is activated by BCL2 proteins, which I haven't told you about yet. Or the extrinsic pathway is activated by DNA damage. So only one of this is true for the extrinsic pathway. Which would you think is true for the extrinsic pathway? Right, cell surface receptors, because it's coming from outside the cell. The rest are intrinsic. Exactly. Okay, so remember I said you get this cleavage of endonuclease inhibitors. So you have these endonucleases that are activated, and you see fragmentation of DNA. Remember that? Well, so this is what it looks like. You get this ladder. So basically, let's say I have cells, and I do a time course where I add some inducer of apoptosis. There's all sorts of drugs to do this. And so at time 0, there's no apoptosis inducer. There's no drug added. And over time, and then I add it for different times, like 1 hour, 3 hours, 6 hours, and 12 hours. And so at time 0, with no drug added, you see no fragmentation of DNA because you shouldn't, right? A happy, healthy cell should not have fragmented. Fragment means broken, right? Fragmented DNA. It should generally be a big blob. Yes. Is that how they make DNA ladders? In a more specific way, I believe. You know what? I have a few different ideas on that, but we should talk later. Because I don't, because now that I'm standing here thinking about it, I was about to say, using restriction enzymes, but that still is not specific. I think they might just put in different sized DNA. That way, because they need to know exactly what size, so it can be a ladder, whereas this is more random. So the way to look at this is large things are on top, small things are on the bottom. So when you have a ladder, that means you have things that are big, smaller, 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 smaller. And so if you've got a lot of nuclease activity and a lot of cleavage, you're going to generate pieces of DNA that are of different sizes. Some might still be big because they haven't been cleaved a lot yet. Some might be smaller. Some might be somewhere in the middle. And so this is your characteristic ladder pattern resulting from DNA cleavage. I recently helped out on a paper where they were looking at the effect of enteroviruses causing apoptosis, and they had this same exact figure. They needed to show that their virus was causing apoptosis in their cell line. Any questions about this? This is something that usually confuses people. So I just want to make sure people are looking at it and kind of understand it, because I like to put things like this on the front. Okay, everyone's happy. Yes?
So you're saying like more time?
Yeah, so usually when you're looking at the effect of a drug on a process, such as apoptosis, you'll do a time course because you want to see a dose response. So with more time, there's more fragmentation, and that's what you see. Right, you see just one big piece of DNA, and then here you see a little fragmentation, and here it gets darker, because actually the intensity of the band means there's a higher quantity of DNA. So the fact that these are lighter in color or in intensity, that means there's less of it. And so over time, you see more fragmentation. Good question. Okay, any other questions? Okay, so here's another assay that people do. There's a lot of assays that people do to study apoptosis. This is a tunnel assay, and what it's doing is it is detecting the DNA ends that are generated during fermentation. And so here you can detect it with dye. It's showing it actually in a frog, development of the frog foot. So that usually have these apoptosis happening between the fingers or digits. Okay, so here's the BCL2 family of proteins. So it's a whole family of proteins, meaning that there are many different proteins that are in this family and they have different functions. And why you call them this BCL2 family is because they share this BH3 domain, so they have some sequence similarity. But you can put them in 3 different categories. They're either anti-apoptotic BCL2 proteins, they're pro-apoptotic BH123 proteins, so they have this BH1, 2, and 3 domains that are similar, or they're pro-apoptotic BH3 only. Either way, there are proteins that are pro-apoptotic and anti-apoptotic. There's a lot of them. And they're in this BCL2 family. Okay? If I ask you a question on the exam about BCL2 family of proteins, I would always indicate whether I'm talking about a pro-apoptotic or an anti-apoptotic one so you can think about it. So don't make the assumption that they're all pro-apoptotic or they're all anti-apoptotic, because students always do that. We're talking about a huge family of proteins with different functions. So the intrinsic mechanism of apoptosis is regulated by this BCL2 family of proteins. Remember, intrinsic, some kind of damage on the inside stimulates apoptosis, and that results in cytochrome C release, right? So in this situation, you'll have some sort of apoptotic stimulus, and these pro-apoptotic BH123 proteins that live in the mitochondrial membrane, they're going to become activated, and they're going to aggregate, and they're going to form a pore. They're going to make a hole in the membrane. And so when you have a hole, you're going to get proteins that live in this intermembrane space. They're going to come out of the intermembrane space. So that's that cytochrome C release. Other things get released also. This is called MOMP. I always call it MOMP. I don't know if apoptosis people call it MOMP. Quote me on that, but it's mitochondrial outer membrane permeabilization. Permeabilize, that means to make whole in. And so this mitochondrial outer membrane permeabilization results in release of cytochrome C from the intermembrane space. So when there's no apoptotic stimuli, you have these other BCL2 proteins, these anti-apoptotic BCL2 proteins that are present, and they inhibit these BH123 proteins from assembling and forming a pore. So that's how the cytochrome C is maintained inside the mitochondria. You don't have the cytochrome C release, and you're keeping apoptosis off. But when you have an apoptotic stimulus, then you get inactivation of the anti-apoptotic proteins, and then you get that assembling with the pore, and cytochrome C comes out. And so one of the other, one of the other ways to keep apoptosis off is through activity of these IAPs, which are inhibitors of apoptosis. Do you think inhibitors inhibit apoptosis or activate apoptosis? I'm just testing you. There's too much time. What do you guys think? Do inhibitors activate apoptosis? Oh God. Sometimes people get so bogged down with memorizing things, they don't think about the words, so I'm just trying to make sure you know that inhibitors of apoptosis inhibit. This is like senior level thinking here. Okay. I mean senior citizen. I love senior citizens. Now I'm gonna be sued by the AARP, something like that. Anyways, IAPs, they bind and inhibit activated caspases in the absence of apoptotic stimuli. So remember, caspases get activated accidentally. That's bad news for the cell. So you have to have this another level of regulation, and that's these IAPs. But notice inside the intermembrane space, you also have these anti-IAPs. So when you do get activation of apoptosis, not only is cytochrome C released, but also these anti-IAPs are released. And so that turns off that system also, allowing apoptosis to proceed. Right, so here it's showing you get the pore forming. Yes? I think you're pretty much done. It's a done deal. Is there rescue from apoptosis? I'm not an apoptosis researcher, so I don't want to say anything definitively. But it's a fairly rapid event. And so I don't know how. I think once you get your executioner caspases active, I think you're gone. I'm pretty sure. But caveat is since I don't work in the field, there can be something I don't know about. Yes?
There was a guy from Optimus in his PhD found evidence of reverse apoptosis.
I've heard that. Yeah, there is someone who studied. There are probably a whole group of people studying this reverse apoptosis, but I don't know anything about it. I should probably start looking into it after in the lecture. Other questions? Anything? Another hand up? Okay, so when you get activation of the intrinsic pathways, these anti-IAPs are released. And this is known in Drosophila, for sure. It's controversial in mammals. But either way, then you get formation of the apoptosome, and then you're going to get activation of your executioner caspases, and then you move forward with apoptosis. Questions? Okay, so this is what I was talking about before with the survival factors. So not only can you induce apoptosis through like this extrinsic pathway where you've got fast ligand, or where you've got the intrinsic pathway where you've got this mitochondrial outer membrane permeabilization and cytochrome C release. Another thing that you'll have, and this is extrinsic also, is a lack of survival factors will induce apoptosis. So for example, with nerve cells, target cells will secrete survival factors, and then those nerve cells are going to compete for them. And the nerve cells that do not receive survival factors or get enough survival factors will undergo apoptosis. So that's another way that apoptosis is initiated. So they work in different ways. So one thing they can do is they might, the survival factors, remember they're inhibiting apoptosis. And so one thing they might do is activate a gene regulatory protein. So again, this is a signaling pathway, right? Because you've got you'll have some secreted molecule from some other cell, bind to a receptor, activate signaling, activate a gene regulatory protein, induce expression of BCL2, and an anti-apoptotic BCL2. So now you've got generation of these anti-apoptotic molecules. When you don't have your anti-apoptotic molecules, you're more likely to activate apoptosis through the intrinsic pathway. You could also have an activation of pro-apoptotic BCL2 protein. So you have your signaling molecule binding to a receptor, and then you'll get activation of AKT. Remember, go back to signaling, AKT. And activating AKT will then result in inactivation of the inactivation of the inhibitor of the pro-apoptotic protein. So then your BCL2 protein will become inhibited, so it won't be pro-apoptotic anymore, anti-apoptotic. So then another way this could work is you might have your survival factor bind to a receptor, activate some signaling pathway, and that will inactivate anti-IAPs. So remember, the anti-IAPs are those proteins that are secreted from the mitochondria, not secreted, are released from the mitochondria into membrane space. When you have that membrane permeabilization, they're going to bind to and inhibit the IAPs. And so here, if you inactivate the anti-IAPs, that allows for IAPs to be active, and those are inhibitors of apoptosis. So in all three cases, you're getting blocking of apoptosis. Spend 5 minutes looking at this, not right now. Spend 5 minutes looking at this figure in the textbook with the figure legends and the text, and it should make complete sense. I know I went through it quickly. Does anyone have any questions about this? Is everyone happy? Everyone's brain dead from advanced studies. I'm so sorry I got you guys after one. Okay. Okay, no survival factor equals apoptosis. So I want you to know that this pathway is not activating apoptosis, it's inhibiting apoptosis. The survival factors are not going to induce apoptosis, right? So don't get turned around with that. Just think it through. Okay, so we're going to end with a quick practice problem. Okay? You're studying induction of apoptosis via the intrinsic pathway. You treat cells with UV. Oh, you guys are just ready to go. That's power through then. You treat cells with UV light, and most cells undergo apoptosis within 24 hours. UV light is the source of DNA damage, just like X-rays. So DNA damage induces apoptosis via the intrinsic pathway. So you can detect cytochrome C release. So here you're looking at cells. That round circle in the middle is the nucleus, and the white spots are the cytochrome C within the mitochondria. When there's cytochrome C release, it becomes this diffuse kind of pattern because it's not concentrated within mitochondria anymore. So you can detect as early as 6 hours post UV exposure, up even 10 hours later, you can detect cytochrome C release. And so your question is whether individual cells are releasing cytochrome C over a long period of time, or whether different cells are triggered at different times. So you use this GFP tag cytochrome C, and you observe the different cells over 24 hours. And so you're supposed to think about which model this data supports. Single cells release cytochrome C over 10 hours, or do different cells, are different cells being triggered at different times? So take a minute to look at it, and we'll go over it. So what this is showing you, you're looking at two things, right? You're looking at 10 hours. So the same picture, 10 hours and then 10 hours, 15 minutes. And then you're looking at another picture of another cell at 17 hours, 10 minutes, and 17 hours, 18 minutes. Does anyone have any brain power to try to think it through? Have any thoughts? How can you know it? Yes. Great. Okay, so the question is, wait, we're just looking at a few minutes. How can we tell if it's over a long period of time? Well, actually, you're looking at 10 hours, and then here you're looking at 17 hours. Post-treatment with UV. And so you detect it as early as 6 hours. And so you're seeing, so the way to think about this is you're seeing this change in just, I'm not going to try to do math, between 10:09 and 10:15, you're seeing that change. And then you're seeing it between 17:10 and 17:08. So if it was all at once, one would expect, if it was all, if it was the same releasing for a long period of time, for 6 hours, when you start to detect it, one would expect that you would see them all kind of looking like this fairly quickly, and it would stay that way, and at 17 hours you'd see even more that were all, right? So I would say different cells are being triggered at different times. Okay, I'm ending with that. Please bring your notebooks to my office today, because we only have 5 minutes and I don't want to end up taking, you can follow me right now. Feel free to leave it with me and take it back later, but if you want to keep it with you, come to my office.
Just put it right there.